Good afternoon, everyone. It is fantastic to see so many of you here eager to learn about infrastructure for developing therapeutics. We have some fantastic speakers today. Really excited to share some of the capability that is available so that people are aware of what's in Australia and what expertise is here, so you don't have to be going to China or Europe or the US to do this kind of work. Before I begin, I want to acknowledge the traditional owners of the lands on which we meet, the Gadigal people of the Eora Nation, and pay my respect to elders past, present and emerging, and extend that respect to any Aboriginal people or Torres Strait Islanders with us today. This event came about from a conversation I had with Heather Donaghy at TIA in early January, where we caught up and had a bit of a chat about how MTPConnect and TIA could work more closely together. Previous to that, I've been having conversations with NMI, ANSTO, a number of the other groups around Australia who provide this kind of infrastructure, who had said, we want to build a bit more awareness in industry about what we do. I knew that ANF had already progressed quite a way down that pathway, and so between us, Heather and I cooked up this idea. What we want to get out of today is build awareness, first of all, of what infrastructure is available in these fantastic organisations. So we want to build awareness of the infrastructure, but also the expertise that's housed within each of them. We want to make you aware of the funding that is available to help facilitate access. And some of the groups have their own programs that they run to help you get in and use the kit that they have. And finally, we want to be able to help you put a face to a name at each of these organisations so that you know in 6 months, 12 months, 18 months, that's the person I need to get in contact with. It's been really interesting to see that some of the groups that are presenting today, we've been able to facilitate connections between them as well. And so not everyone knew each other. I think all of the NCRIS teams knew the other NCRIS teams, but getting ANSTO and NMI involved in too has been another outcome that we're really quite excited about. For those of you who aren't familiar with MTPConnect, we are Australia's Life Sciences Accelerator. So we began life in 2015 as the Industry Growth Centre for MedTech, Pharma, Biotech and Digital Health. And we're funded by the Federal Department of Industry, but operate as an independent, not-for-profit company distinct from government. And basically what we exist to do is help build the sector in Australia. We're a national organisation with people based in Sydney, Brisbane, Perth, Adelaide, Melbourne and Jindabyne. And Elizabeth is with us today from Jindabyne. And while we have our geographic responsibilities, we don't work to do things to the benefit one state to the detriment of others. We're about leveraging deep connections in each state to the benefit of everyone, and that's how we work. Going forward, we've also been named as one of the industry partner organisations for the Industry Growth Program, and I don't come up with these names, I apologise for the very similar sounding names, but that's what we are. And we're going to have Tony from the Industry Growth Program join us at about 3:30 to talk about that opportunity. I want to say a huge thanks to a couple of people. First of all, the organisations who are presenting today. We really appreciate you coming along and talking about what you do and building awareness. We also really appreciate the promise that you're going to keep to your allotted time, because we're going to have to push you off the stage if you don't. I also want to say a huge thanks to the companies who've agreed to come and talk about the work that they've done with the organisations. It's time out of your day as busy founders, and we really appreciate you doing that. I want to say a big thanks to Heather, because without Heather pushing us, we would not have done this. So thank you for that, Heather, and look at what we've got. This is great. Most importantly, I want to say a huge thanks to my brilliant colleague, Elizabeth. And without Elizabeth, this event would not happen. So Elizabeth has been with MTPConnect from the very beginning. I have the privilege of working with a lot of brilliant people at MTPConnect. Elizabeth is the glue that holds the company together. So please just join me in giving a quick thanks to Elizabeth. And I also want to acknowledge my colleague, Dr. Manna Liao, Director of our TTRA program, who's also with us today. Thanks, Manna, for joining us. With that, I'm going to pass to Heather, who's going to chair this session, and we'll kick things off. Thanks, everyone.
Thanks, Duncan. And thank you for already giving us an extra 10 minutes of time. So, as Duncan said, we cooked up this plan to have everybody here, and I'm really pleased to see so many people in the room. It's really great to be able to showcase everything that's available to Australia, in Australia. So I am going to move on to, this is the agenda. Okay, so as you can see, it's a pretty full program, 10 minutes, 15 minutes for each of the infrastructure providers to speak, and then 10 minutes for a case study on how an industry partner, how an SME has used that facility or that capability in order to progress their program. So we're going to start with Andrew Gilbert from Bioplatforms Australia, the CEO of Bioplatforms Australia, who'll talk to us all things omics.
Thank you, Heather, and thanks to MTPConnect for organising. It's really appreciated and it's lovely to see some friendly faces. I'm Andrew from Bioplatforms Australia. We're one of the health-related NCRIS capabilities, although we also work on everything from koalas to wine characteristics, but there's a bunch of others here today and some additional ones, ANFF and microscopy and EMBL Australia and so on, who have quite a lot to do with the NCRIS Health Group, but as a collective we provide a pretty comprehensive set of capabilities and can help steward projects that might require a multiple set of these activities on behalf of you through the complexity that is NCRIS. You'll probably see this slide 5 times, I reckon, today. But Bioplatforms itself, we have a variety of different functions. Historically, we support enabling labs in the spaces of genomics, proteomics and metabolomics. More recently, we were very fortunate to secure some substantial funding from NCRIS and the Commonwealth Department of Education to build a synthetic biology capability, and Tom later will give you an insight into that synthetic biology capability and how it has supported his company. These disciplines, of course, are increasingly digital, and we have a very large digital capability that's freely available to everybody in the country, providing access to software, computing environments, dedicated access for life science into Australia's 2 public high-performance computers, of course digital skills that can help you adopt and use these technologies for your own purpose. We run a variety of what we call framework initiatives, and really they're public collaborations around themes of some national significance, probably less interesting to this audience here today, but think things like melanoma, where we've brought together all of the significant melanoma clinicians, researchers and geneticists around the country and built very large scale, population scale melanoma studies that of course have then informed clinical trials, use of very expensive drugs on PPS, and contributed very significantly to global pan-cancer efforts. That particular study was led by amongst others, the 2 Australians of the Year. Not that we had much to do with them becoming Australian of the Year, we're just very fortunate to have leadership of that type of calibre supporting us. Finally, more relating to today, we have a dedicated effort around partnership in support of impact and commercialisation. Indeed, we have a leader in Bioplatforms called Vicky Snelson, who should be here talking today, but she's in fact off running commercialisation training and workshops in Perth this week. Everything we do, of course, is in support of impact. It's medtech today, but of course we do a lot in the agri-food space and environmental biodiversity realms as well. A more detailed list of the types of capabilities that we support. We take great pride in having capacity and currency in just about any technology that you might require in biomolecular analysis around the country. If we don't have it, we could get it, but we need to be made aware of where those gaps are and the types of demands that you have, and it's our job to ensure that we have these capabilities at the currency, the quality system level and so on that in fact you require. This network of capability is delivered all around the country. Here in New South Wales, though, we have significant labs at APAF at Macquarie University, the Australian Genome Foundry, and the Ramashotti Centre located at the University of Sydney, but they're but 3 single laboratories that are a set of integrated capability that permits access to a broad set of laboratories and differential expertise around the country. I won't go through every site we have, but you can find on our website there the research platforms pages, the types of information you might need to access the facilities that we have. Please feel free to contact me directly though to make introductions as you see fit. We do think though laboratory access, performing assays, supporting product development is only a dimension in ensuring that there's an environment for success for startups and SMEs within Australia. And so we've gone about our business over the last few years building an ecosystem and network of capability and expertise across not only the technology platforms I've mentioned there in the dark blue centre circle, but a whole lot of peripheral capability and infrastructure that's necessary through product design, connectivity into regulatory affairs and freedom to operate, support of ethics committees across our universities to reduce the barriers of entry of SMEs as they come into their product development pipelines, and of course more and more connected into commercial ready environments. And in fact some of our partners here in D4, David Barter up the back here, you might like to go and introduce yourself during the break. We're working very closely in trying to help the likes of you through these types of environments. And finally into translation, where we're partnered very closely with some of our NCRIS peers, notably Therapeutic Innovation Australia and Scale Up, Phenomics Australia in providing some preclinical models, and so on and so forth, to ensure you have a broader set of capability available to you, understanding that it isn't just what we do that will help you succeed, but of course everything the National Research Infrastructure Set can bring to bear to your challenge. Our approach, we act almost as an accelerator in our own right. To give you an idea, we supported over 4,000 discrete clients last year, 20,000 projects, and so that's a very large body of work across the country, and that really provoked us to set up almost an internal accelerator that aspects of it might be interesting to you. First of all, it's opportunity identification. There's so much great science done in Australia and so much of it is lost between the cracks in academic environments. And as I said, we have a whole network of staff, almost 350 FTE around the country now, who are being trained to identify those great opportunities and help triage and identify programs that might come to us via an EOI, where we can sit down and meaningfully engage with these opportunities and the custodians of them in how we might assist. Secondly, of course we bring infrastructure to bear on the problem. We have significant network capability. All of our facilities around the country actually have access to incubation space as part of our program design. In fact, some of them did not, and it's probably a detail you don't need to worry about, but we insisted under contract that they did, and we were shocked and pleasantly surprised at how enthusiastic the academic sector was around the country to embrace this idea, and where they did not have space, they created it, because of course universities themselves have increased targets and measures and opportunities around commercialisation space. So it's an inbuilt facet of how you might engage with us. I've mentioned legal and regulatory support. We work very closely, have regular structural meetings, for instance, with the Office of Gene Technology Regulator and the vet and medicine folk relating to livestock and agriculture. We're building an enhanced set of mentors around the country, and indeed are taking steps to try and access very significant networks of mentorship out of the west coast of the United States through some partners we have and some direct contacts that Vicky from our team has built over the past year or two. And of course, it's that mentorship in commercialisation that I'm probably talking to the wrong audience here, but there is a significant shortfall of that in the Australian high-tech ecosystem. We think it's just an invaluable contribution that is reasonably easily met. I guess when it comes to the bit that many of you do care about, how to get money. Of course, we have finance of our own that I'll come to in a moment. We're very system aware though. We keep full registers of the non-dilutive type of grants, public-private partnerships, state and Commonwealth that are available to you. And we've partnered very successfully actually with pre-seed, startup and SME companies for 10 years now. Some of those outside of the world of medtech. For instance, we seeded some laboratory activity. We're able to co-invest in a renewable energy agency grant for a particular alternative hydrogen program. That took that organisation from being a laboratory function to a real company capitalised with $1.5 million of non-dilutive funding, and they've gone on and been through a Series A and secured $6 million worth of support. Of course, that was them doing that, but we were able at the very high risk, early stage, bring to bear this overall program of work, some modest seed funding we had, and supported a grant and then subsequent commercial, not quite success, but commercial empowerment of that particular opportunity. Hygiene is the name of the company. Of course, much of this is aimed ultimately to bringing mature risk investments and venture opportunities to investors. And we are doing this more and more. Warm introduction to a network of investors that we've built and met at functions like this around the country for many years now. And finally, we have our own pre-seed funding actually. Funding, I should add, that is not NCRIS support. We hold certain types of monies that are intended to provide venture-like funds, often under things like safe notes for high-risk opportunities, where not only can we bring resource, but we can bring this ecosystem of opportunity to assist really those startups, sometimes even pre-startups, into an arrangement where they have that 1 year or 18 months opportunity to go away and succeed in their own right. We're prepared to share in that risk. Tom, who will talk on behalf of Number 8 Bio in a moment, is an example, I should say not a medtech example, but all the same, swap out Tom's technology for what interests you and you'll get the idea. Tom has actually been through, to a certain extent, this type of journey. I'll let Tom tell his own story, but we were able to fund under safe note, seek some co-investing private finance in support of floating Tom's company that then gave them a runway to meet milestones and secure the opportunity for them to scale out as a company. So that's our little story, all within 15 minutes. The last person who isn't here today, they're actually off at Bio, Michael Dobby at Phenomics, Stuart Newman at Therapeutic Innovation, Heather, and Bioplatforms, all co-employer staff. Remember John Pariso, and it's his job to help you. I'll help you too, and Michael and Stuart and Heather, everybody will help you, but John is absolutely a point contact for this type of activity. So, thank you so much.
Thanks, Andrew. And I should have said earlier, we're skipping out on questions so that we've got enough time to get through everybody, but there will be time at afternoon tea and also in the networking at the end to ask questions of everybody, and you now have contact details and a face to talk to people. So next up, I would like to introduce Tom Williams, who's the founder and CEO of Number 8 Bio, who's going to talk about how he interacted with Bioplatforms.
Thanks, Heather. Thanks, Andrew, for the snippets of intro on Number 8 Bio. So we've benefited really profoundly from interactions with Bioplatforms Australia through both direct investment that they made in my company, Number 8 Bio, as well as through incubation at one of their facilities, the Australian Genome Foundry. But I thought the best way to explain the extent of that impact was actually to give a brief background on my company, Number 8 Bio, and what we do. So I'm Tom Williams, I'm the CEO and co-founder. And at Number 8 Bio, what we do is incredibly simple. We stop cows making methane and increase their productivity, and we do that with feed additives. So this is a really important problem space to work within, because methane emissions from Earth's 2.6 billion cattle and sheep account for about 6% of all climate change that we cause as a civilization. On top of that, those methane emissions are really short-lived and potent in the environment. So they're about 80 times more potent than carbon dioxide, but they also break down over around 20 years. So to make a rapid and profound impact on climate change, methane emissions are arguably the best place to be. The beauty of the problem space, though, is that it's not just an environmental problem, because all of those methane molecules that are belched out of our cows actually represent wasted energy from the animal's feed. So in theory, if you can shut off those emissions, you can unlock 10% more energy from the feed and give the farmer a financial incentive to adopt a climate technology like ours. And then on top of that, the world is changing. We have governments, banks, corporates all starting to be required to report on both their emissions and the emissions within their supply chains and with their customers. So this type of technology, even without the productivity benefit, definitely has a future. So what we do at Number 8 Bio is to make the most scalable, the most affordable, and the most effective methane mitigation feed additives for cattle and sheep. And the key unlock to this entire market and the core of what we do is to make productivity happen for farmers. So either they have reduced feed intake for the same amount of meat or milk, or they have more meat or milk for the same amount of feed. And we've been able to do this through deep scientific expertise and a novel approach to the technology space, where instead of picking like a single technological intervention and testing that until the cows come home, we've taken a more systematic approach, which is very common in synthetic biology actually, where you have as many shots on goal as you possibly can, explore as many variants of the product and the technology, because we are trying to engineer an incredibly complex system in the rumen, which has all these microbes in it. So we developed a high throughput screening process that allows us to test 90 product variants per week, and that's the Australian Genome Foundry, which Bioplatforms Australia funds, has been critical to that, and I'll go into a bit more detail on how that works soon. The other critical component to our success has been our team. Again, we are somewhat different in this space in that we're led by microbiologists and scientists, and we are able to deal with the deep complexity of the rumen environment. So the expertise also spans growth and sales, agricultural product development, and metabolic engineering. So how does the rumen work and what does our product do? Well, the rumen is a heaving bag of billions of microbes that convert plant material into nutrition for the animal. Cows can eat grass and cellulose because of the microbes in their rumen, not because of anything that the cow does that's special. So our products work by being mixed into the animal's feed. We need about 25 grams per day. This consistently shuts off methane emissions. We've now done 2 live animal trials where we've seen a 90% decrease in methane emissions using 2 independent product lines. This frees up metabolic energy in the form of hydrogen, which we can then divert towards productivity using a special mix of prebiotics. And we have patents that cover all of this. The other key thing that differentiates us from other companies in this space is that we've actually developed different technologies for different market subsections, because interestingly, the customers care what is in the product and how it works, and they have quite different requirements for how it's deployed. So we're working right across beef and dairy and grazing systems, as well as making a natural version of the product. And each of these actually has different inhibitors and different cocktails of what we call productivity modifiers. it's really important that we have this diverse product spectrum because we have to provide value to customers at two different ends of the supply chain. So with farmers, we have to give them a productivity boost, and then with corporates, we have to really decrease their emissions. And the different product classes are important for that. So as an example of our commercial traction, we're working with a large Australian dairy company that wants to roll our products out across their entire supply chain. And we're also working with a really prominent burger brand supply chain manager that has similar intentions. So now we get to how, like, the history of our company and how BPA's impacted us. So a bit of a historical overview here. We founded the company in mid-2022, just my co-founder Alex and I, and Bioplatforms Australia actually put in our first check of $250,000 through a safe, and that was really important because at the time I was an academic. I had tenure at Macquarie University and needed that vote of confidence in part to take that leap and essentially leave academia, take on the legal cost to found the company and get going. So that was a huge impact on Number 8 Bio in itself, and the confidence that signaled to other investors was really important as well. So from there, we went on to do an accelerator program run out of UNSW called Founders 10X, and through that we also got a voucher from Bioplatforms Australia of $30,000, which I think is probably all gone now, but that's been, again, a big help to help increase our runway. From there, we attracted another 1.5 million in pre-seed funding through safes from both venture capital and other angel investors, and then used that to hire some more staff. So here's my co-founder Alex and I and our first two scientists, Dom and Silas. And from that point on, we started incubating within the Australian Genome Foundry, which is a world-class synthetic biology facility equipped with all sorts of high-throughput equipment. And that turned out to be actually essential towards our success because we hit a critical bottleneck really early in the company. We have to test our product variants using a system called in vitro rumens, which involve getting rumen fluid from a live cow, bringing it into the lab in a bottle and feeding it with hay. The microbial community then breaks down the hay and makes methane in the headspace of the bottle, which we can then measure using analytical chemistry. So we originally were outsourcing this at a different university, and they were going to charge $10,000 for 30 samples, and it would take 4 months. So we thought, well, we're dead if we do that. So we went about sourcing our own rumen fluid, developing our own in-house in vitro rumen testing system, miniaturizing it and automating it using equipment that BPA had funded within the Australian Genome Foundry. So we now have, we think, the highest throughput in vitro rumen testing capability on Earth, and we can do 90 samples per day using partly automated gas chromatography mass spectrometry. So in the last 12 months, we have screened well over 2,000 variants of our product, and I guess whittled that down to a few that are now going through live cattle trials and having great success. Off the back of that, we raised a large seed round, which isn't public yet, and I can't tell you how much it was, but we are now using that to build a manufacturing facility. So this is our warehouse in Lane Cove West, 500 square metre space where we make the product that goes into the trials and into early commercialisation. It has a few more things in it than in that photo, but not too much. I think it'll take some time to fill it up. And we have expanded the team from 4 to 10. So we now have a maturing capability within the company to both do the science and commercialise it. And like I said, we've had already 2 successful cattle trials this year, and we have about 8 more booked, which follows this shotgun approach to product development that we have. So while I have your attention, we are looking for partners that are animal product brands, farmers, and Series A investors to help us grow. So please feel free to come and see me in the break or to email me afterwards. Thank you.
Thanks, Tom. It's a great talk. So we're going to move now on to the Australian National Fabrication Facility. And we have Oded Van Ham, who's going to talk to us about the capabilities there.
Thank you. Good afternoon. I'm Oded Van Ham. I'm a client engagement facilitator for the Australian National Fabrication Facility. Established 17 years ago by NCRIS, and you're going to hear NCRIS's name mentioned a few times, so Andrew did already. Let me just explain that NCRIS is a federal program started 17 years ago, I believe. It's a National Collaborative Research Infrastructure Strategy. It's designed, it's funded to bring resources into Australia that's of benefit to a lot of Australians, but no single organization can typically afford to kind of purchase it or to acquire it. So NCRIS funds us to bring these resources in and make it available to everyone. Now in our case, in AMP's case, it's, we provide the R&D communities with open access to advanced manufacturing capabilities. We're nationwide. We're located across the country in 21 hubs, which are part of 8 specialist nodes. Now, AMP supports innovation, supports innovation in all sectors. Startups to multinationals. We have a particular strength in medtech. We help companies develop new or innovate their existing products and overcome manufacturing and technology challenges. So we provide access to leading edge manufacturing facilities and expertise. Now let me kind of focus on that a little bit more, because a lot of you might be thinking, well, there are a lot of companies, a lot of places that I can go to for advanced manufacturing. A lot of service providers do advanced manufacturing. I can go to maker spaces and have access to some pretty cool equipment. That's all true, but what we focus on is the very cutting edge. Like when we focus on the equipment that if it exists elsewhere in Australia, it's typically only at a multinational large corporation. If it exists at all within Australia, often the equipment that we have, we're the only ones that offer access to it. And we're the only ones that have it. And let me also focus on the expertise aspect. So, we've got, over the 17 years that we've been around, we've spent $500 million on equipment. So that'll give you a sense as to kind of how cutting edge the equipment is. But equally important is the expertise that we provide. The engineers that work on the equipment are world class. They've worked they're able to get the most out of the equipment, having worked on a diversity of projects across multiple sectors. So they're really just amazing people to kind of support innovation. And that's what we're here for. We're a not-for-profit organization really designed to help support innovation across Australia. And because of that, we try and keep our barriers to using us as low as possible. We want people to use us. We want people to use us and innovate, try new things. So there are multiple ways of engaging and for your research, development, and commercialization needs. The first is we're open access. So all this amazing equipment that we have is available to anybody to use. You can come and book the equipment and use it yourselves. You can find out what we have and come and use it yourself as an extension of your in-house capabilities. You don't know how to use it? We offer training. Some people prefer not to do it themselves, in which case we also offer fee-for-service. You provide us with the specs of what you're trying to build and let our team make it for you. We also offer project advice and consultancy. Now, again, being a not-for-profit, really trying to encourage you all to use us, we're low cost, below cost recovery, so we're inexpensive to use, and we are also IP agnostic. So we make no claim on the IP that you develop with our facilities. Now, one obvious question is, well, what are your capabilities? Well, we have over 500. It's the hardest thing for me to kind of convey to an audience because you're not going to be able to read it all, but it's trying to convey the diversity of capabilities that we have. It's everything from micro nano fabrication to micro milling, precision laser cutting, to injection molding, to fibers and textiles, to fiber optics, and it goes on and on and on and on and on. My suggestion to you is don't try and figure out what we've got. Just come approach us, ask us if we've got what you're looking for, even more generally ask us, well, I'm trying to solve this problem. Do you think you might be able to kind of help find a solution? Now, this being a New South Wales showcase, I thought, okay, I'll focus primarily, I specifically have a slide focusing on what's available in New South Wales. I talked about 21 locations across Australia. We've got 6 hubs within New South Wales, within the state. They're part of 3 of our nodes, ANF New South Wales, ANF Materials, which focuses on new materials and also 3D printing with a diversity of materials that aren't typically known to be 3D printed, and OptoFab, which is, focuses on optics. Capability highlights include micro and nano fabrication, advanced material processing, 3D printing, including bioprinting, and on characterization. And these can be applied to rapid prototyping, PCB prototyping, microfluidics, standard and quantum sensors. Again, this is just the highlights of what we do in New South Wales. Again, the list goes on and on. Now, I mentioned before I'm a client engagement facilitator, so let me explain what that role is within the ANFF. Now, I talked about we've got over 500 capabilities. We've got over 500 capabilities spread out across 21 locations. We don't typically have much duplication from one location to the other. So it can be a challenge for somebody looking to engage with us. Who do we go talk to with so many capabilities, so many different locations? Who do we talk to? So the client engagement facilitator is a single point of contact for you. You come talk to us, and we will concierge that request and find a solution for you. Now, my goal, our goal as client engagement facilitators is not to really kind of drive up business for ANFF. I'm a solution finder. I engage with people within industry that have a challenge in manufacturing or technology, and I look for a solution. I'm equally happy if the solution is outside of ANF as I am if the solution I find is within ANF. And I should also mention it's taxpayer-funded service, so there's no cost to you. Now, the engagement process is pretty straightforward. I'll set up a meeting with you, find out what you're working on, what the challenges are you're having, again, like with you might be working on, you might actually already know, well, I want to use this particular kind of capability that I think ANF would have, and then it's easy for me. I just say, okay, well, yeah, this hub are your options. Let me connect you to the one that's most convenient. But more typically, people will come to me and say, okay, this is what we're working on. We're having a challenge with this component. We're having this we've got this hurdle with our technology that we can't kind of overcome, or we're trying to build something that others have told us can't be built because it's too small. It's got like features that are in a micron nanometer scale. Well, then they've come to the right place because I can find, I'll do my best to find a solution for you. But that's typically what happens in the first stage. I find out at a high level at least, what you're working on. Then I engage with our network to try and find a solution within who's the best suited to talk to you. And as I said, sometimes the best solution isn't within ANF, but our kind of teams are so well kind of versed in these technologies that if they don't know, if they can't do it, then they can often refer you to somebody else that's a good person to talk to. So then, once I identify the best solution for you, I make the connection. I'm kind of a matchmaker. I sit in through the first call. You can then talk in more detail, the nuts and bolts of what you're working on, and talk towards starting a project. And my role kind of typically will stop there, but I'm still available for any kind of needs you might have with that project or any further projects you have down the road. Now, we've got a new initiative, ANFC, that will be of interest to many of you. ANFC is ANF's funding. So we've got a grant that we offer. It's non-dilutive pre-seed fund up to $120,000. Invested in early stage entities. So I see phones coming up to take pictures. Yeah, notes being taken. Why are we doing it? It's to reduce the risk in preparing for a professional investment round. Now, the way it's structured is there's gate one that's up to gate one that's up to $20,000, and gate two that's up to an additional $100,000. So you can, as I said before, get up to $120,000. Now, these funds cannot be used for ANF use, but rather what the funds would be used for is decided based on consultation with our ANFC team and of course you. What would be of most benefit to you to move your organization, your startup further down a commercialization journey? So through that consultation, we decide, okay, well, that's the best use of the funds, and that's through that agreement we will fund that aspect. Oh, an obvious question: what are the criteria for ANFC funds? You need to be a client. It's pretty straightforward. You need to be a client. You need to have developed a substantive part of your technology with us, and you need to be in need. So if you've got a lot, if you're flush with cash, well, then you don't probably qualify. That's not usually an issue. Now, I guess that's it for me. That's my contact details. So as I said, I'm a client engagement facilitator. I encourage anybody that has a manufacturing or technology need, reach out to me, tell your friends. If you have any questions about what we do, I'm available during the break session afterwards. Matt Chong, raise your hand. Matt Chong's a colleague of mine, also a client engagement facilitator. We're available. Naveen's in the back there. He's still a client engagement facilitator for the next 24 hours. Unfortunately, he's leaving the organization, but you can talk to him too. But yeah, feel free to reach out to me today or beyond. Thanks very much.
Thank you, Oded. I should just mention that all of the contact details will be shared with the audience afterwards, so for all of the contact details of the speakers and how you can approach us. And also at the break, there'll be slides up showing everybody's names and contact details. So next we have Stephen Vasilescu from Neogenics, who's going to talk about how he has interacted with ANFF.
Thanks, Heather. Okay, thank you to MTPConnect, and thank you very much for AMP for inviting me to kind of present to you as a bit of a case study. We have benefited fantastically by working with AMP, both in a manufacturing sense and in a funding capacity as well through the AMPC that Oded mentioned. So what I might do is I'll talk to you a little bit about what Neogenics does, what our journey's been, and then how we've engaged with AMP for manufacturing and for the funding that we've received and how we've used it. So Neogenics is a medtech startup developing technologies and software for sperm selection in IVF. So we're working in the fertility space. So 1 in every 6 couples are infertile, 1 in every 6 of reproductive age. And what a lot of people don't realize is that while we have IVF to treat infertile couples, 78% of IVF cycles fail. That's a huge, huge number. And a big part of that really is the technology that we're using to treat infertile couples. And especially when it comes to selecting sperm for IVF. We see a lot of innovation in eggs and embryos and drugs for women, but not a lot has been done on the male side and selecting sperm for IVF, which is a bit strange when you think that we're sperm are contributing half the DNA for an embryo, as well as some essential proteins and microRNA and all that sort of stuff. So Neogenics' mission is to obviously improve IVF success rates through superior reproductive technologies. So more babies, basically. And we're doing that through a hardware and a software solution. And AMP has really helped us with both, but particularly in the hardware side of things with their manufacturing capabilities. So the areas that we're looking at, about 90% of men have motile sperm in their semen, means that they have sperm that can swim. And about 10% of infertile men have no sperm whatsoever and go through a surgical process to find sperm. And essentially we have a solution for both. So we're catering for the whole infertility market and male infertility market. So when it comes to sperm selection for IVF, clinics today actually don't have a reliable method for selecting sperm. They can't select sperm safely. They use a centrifugation-based process which can damage the DNA of sperm. I always use the analogy that, what clinics are doing now is they're spinning sperm at about 500 Gs in their centrifugation. I use the analogy that astronauts, I think, pull about 3 Gs. Fighter pilots pull about 9 Gs, and we're spinning sperm at 500 Gs. So you can imagine that they're not having a very good time. And what that does is that can lead to poor embryo quality, lower implantation rate. The big one is miscarriage. You can almost triple your chance of having a miscarriage if you're using DNA-compromised sperm. In some forms of IVF, such as IUI, where we're putting sperm into the uterus, that can be a 5-fold risk, 5-fold increase in the risk of miscarriage when you're using compromised sperm. So for our hardware, we have a sperm selection device that replaces the conventional methods by mimicking the female reproductive tract. So we've got specific geometries that try and trick sperm into thinking, oh, I'm inside the female body, I know where to go and what to do. So it essentially becomes like a sperm Olympics in a sense. For our software side of things, for these severe male infertility cases where men have an IVF surgery, once we get tissue from the testes into the IVF lab, embryologists have to spend up to 12 hours looking for sperm manually. What we've developed is an AI platform that is a sperm identification tool, and it rapidly identifies sperm from this surgical tissue, and we're reducing the sperm search time by about 70% in clinics. And actually, this is the first presentation I've given where we've got exciting news. We got our first pregnancy last week from this AI. Thanks. So we're very, very excited. This was a go, no go case basically where our sperm found 15 sperm, where 2 unassisted embryologists in a side-by-side study only found 3, which meant that we got to fertilize all of the eggs that the woman was able to give. We got 2 embryos from the AI-assisted sperm, and as of last week, she's pregnant, which is fantastic. And I have to say, without the funding support from AMP to kind of push these initiatives and give us the hardware and the computing power to deploy to these clinics, we wouldn't have been able to run this study pretty much. So the value from these 2 products is on the sperm selection side for the device, we're giving healthy, undamaged sperm in a quarter of the steps and about half the time. So there's a big workflow improvement as well as the quality benefit that we're getting from selecting better sperm. The AI, like I said, is reducing sperm selection time. It's a big difference between finding sperm in the first hour versus finding sperm at the 6th hour or 10th hour, right? And it's capable of plugging into existing microscopy setups within IVF clinics. We're now deploying that to the UK as well, and we're going to the US, to Singapore, and Dubai, and Hawaii next month to demo this AI across all those different sites. But now, working with AMP more specifically, we've received tremendous support even before we started the company 2 years ago. During my PhD, which was developing most of this technology, we were working with AMP from the very, very beginning. They were giving us design support on our microfluidic devices, and we did early trial manufacturing with AMP in South Australia, which kind of paved the way for design iterations to the device that we have now. The AMP South Australia in particular have been providing precision manufacturing, machining for us, and we're still working with AMP today. These are new photos from the last week from some of the precision micromilling that AMP's been doing for us. I actually talked to some companies out of the US and China saying, I need these resolutions and I need these aspect ratios, which I already knew AMP was giving us, and they told me I was crazy. They said, oh, there's no way we're going to give you this, or it's going to cost you several hundreds of thousands of dollars to build this. And I go, well, I have someone, so I don't even need to. We don't need this conversation. But the reporting that we're getting out of AMP is also top-notch. So I think what Oded mentioned, which is the cutting edge of manufacturing and the capabilities that AMP have, they're constantly, throughout our engagement, been rolling along and upgrading their facilities as we've been engaging with them. So we've been able to push the boundary with our manufacturing as well to replicate those features and replicate the female reproductive tract, which has been fantastic. So this is the core part of our team. We have some fantastic fertility specialists and doctors. We work very closely with Virtus Health, who are IVF Australia, Melbourne IVF. They're the largest service provider of IVF in Australia. I think they're in the top 5 in the world as well, so they have clinics all throughout Europe and Southeast Asia, which is fantastic. And I promised that I would mention the AMP South Australia team because we work very, very closely with them. Simon and Mark have been absolutely fantastic supports for us. I had a call with Mark yesterday, and he said he's losing hair over the project that we've been doing. So if that's not a commitment to how hard he's working on our project, I really don't know what is. But our traction to date, we've done, as I mentioned, side-by-side studies showing that our devices are better, our AI is better. We've done trial manufacturing. We've continued to do trial manufacturing with AMP. I think I mentioned we've also got the Gate 1 and 2 funding support from AMP, and we're continuing to use that, which is terrific. We raised a little bit of money last year. We did the Health 10X program, so Virtus and Medical Angels invested in us, as well as the UNSW Founders Network and TGI. The next stage for us, as I mentioned, is we're moving across different areas. We're trying to expand not just our capacity in Australia, but also we're seeing interest from Europe and Southeast Asia and in the US. We have a base now in the UK. We're working with Burn Hall, which is actually the first clinic to perform IVF in the world. So there's a fantastic history of IVF there that we're working with. And I think we've got a demo next week where we're leaving our AI with them. So now we're raising to scale up our manufacturing, get ISO compliant, and to run a clinical trial with our partners in Virtus Health and move towards a TGA submission. So if you'd like to come talk more about our engagement with AMP, or you'd like to come talk more about sperm, I'm always happy to talk about sperm. Come meet me in the networking session. Thank you.
That's great. Thank you, Stephen. Great story. Not many people say that their work is actually making babies, so that's a really great story. We're going to move on now to Phenomics Australia to understand a little bit more about, let me just double check that I'm telling you this right. Yes. A little bit more about Phenomics. I've got Michael Dobby, who's the CEO of Phenomics, come to talk.
So thank you very much to MTPConnect for the opportunity to put us on your map. And I hope that over maybe the forthcoming months and maybe years, you can come and be part of our map. I've got a few books, which is our impact book, just floating around. It's also available on our website. But we'll really be happy to have the opportunity to talk to you today or talk to you in the future about how we might be able to help find solutions to your problems. And I think you're starting to get a flavour for what NCRIS is. We're yet another one of the NCRIS capabilities. The program itself is about 24 plus projects. A number of them work together in the health space or in the therapeutic space. But we kind of do a lot of things very much collectively and collaboratively. So today what I just wanted to give you is a little bit of a flavour of what we offer through Phenomics. So that's some of the other Phenomics that you didn't hear about from Andrew earlier today. But we work with AMP, we work with TIA, we work with many other facilities. So I think if nothing else, what I'd like you to understand today is no matter where you first contact NCRIS, be it through us or through one of the other projects today or elsewhere, we collectively will find a solution for you. We want to understand what your problem is, and we want to get you to that solution as quickly as possible. So today, it's just going to spend a few minutes just talking about giving you very much a flavour of what we do broadly. Our applications are diverse. Our technology is fundamentally models, making model systems. For the room today, they're preclinical models, but not necessarily exclusively for health applications. So where we cut our teeth was 18 years ago, as we were founded as part of one of the NCRIS founding projects, was we were very, and in fact we still are, very early stages of understanding what the function of every function of every gene is. And so we very much are still part of that activity, but also much more applied activities in trying to now take those fundamental understandings of what genes do to understanding what happens when they go wrong, or conversely, and probably more helpfully, which are the genes that keep us healthy, not only which are the ones or the mutations or alleles which make us sick. So we're very interested in the interactions between genes and environment, the applications, of course, of thinking about new diagnostics, new therapeutics, and both for clinical and veterinary applications. But ultimately what we are is a bioengineering outfit. So we make things to order. There are cells, there are mice that you can buy off the shelf, but often they're not what you need to study what you are interested in, or to design a solution for the problem that you're interested in. So we're very much designing tools using today's latest, sharpest scissors. You've heard of CRISPR, taking advantage of a lot of the other capabilities which are available in Australia. And increasingly moving into engineered living materials. So that's where some of the touch points that we might have in the future with ANF in terms of bringing material science and biological science together. So that's where we sit broadly. But specifically, we work collectively together. Second time you've seen that, keep count. So again, no matter where you touch us or where you make contact with us, I will be able to find that solution for you. If you're familiar with the technology readiness levels, where we sit is really all the way from the very earliest stages through to TRL 5. So the whole preclinical space we represent. If not directly, we can certainly make available through our partners and through our associates access to this. So again, this is federal government supported, not just federal government. You will hear in the next talk about some state government funding that's just been announced. So we work very much in bringing government funding, be it state or federal, and our institutional expertise to this, to the need for designing, building, testing, and learning from what we make. So this is applying the bioengineering rhetoric to what we do in terms of making models and analysing models, helping you to design the very best model, and then actually providing that model for you. Or you're bringing materials to us, be they animals or cells or reagents, and us being able to customise, sharpen them towards where they need to be to solve your problems. So very much into the building your engineering, 3D bioprinting, again, in a different environment to what ANF provides, and high throughput, so miniaturisation, high throughput screening. The test is really the phenomics part, so the phenotyping part, characterising these model systems, designing assays. Often you might have a very good model, but you might not have a very good assay, and ultimately it's the assay that is going to allow you to critically interrogate the biological pathway or look for their biological solution. And we are also very invested in not only what we build and the data that we make, but also generate, but also ensuring that that data is well curated, that it has its full life, that it is connected to other data, and also for the cells and the animals that we make to also ensure that they are available. Now, I know most of you here are not academics, most of you here have commercial interests, and again, we have no interest in the IP that's generated through the projects that we support. We actually have no interest in the animals or the cells that we make customised for your needs. We can curate them for you, but that's not necessarily the case. And so we can keep information, keep resources, biological resources, under embargo, under high quality conditions for you on your behalf. and not necessarily make them publicly available or discoverable. However, with the academics projects that we support, we do encourage them and work towards public sharing of all those resources that have been ultimately publicly funded or co-funded. So I spoke about a map. This is our map. This is our Australian footprint. Up in the top right there, you can see the kinds of the classes of facilities that we make that are colour-coded through biobanking in vivo, which are mouse models or fly models, Drosophila models, our in vitro engineering capabilities, and then our functional genomics and high throughput screening capabilities and our histopathology capability as well. And then those capabilities are spread throughout all of our partner organisations, which are 50% universities and 50% medical research institutes around Australia. You can see them all there. There's information about that, of course, on our website in our book. And also I'm very happy to have these slides shared too. So if there's an opportunity for that, then please delve into the details there. I won't dwell on this slide, but I'll just say that these are effectively the specific capabilities and the specific technologies and the specific applications that we provide through our partner organisations. So how can you access that? So we have an open call for coming and talking to us, but we also have twice a year a voucher scheme that we run in concert with Therapeutic Innovation. In fact, Therapeutic Innovation Australia have been running this voucher scheme for a number of years. We're now coordinating with the voucher scheme in the last couple of rounds to share the promotion and the assessment and the monitoring of the applications and projects that we support, and also working with ANSTO in the current round. So this is a way to access up to $50,000. We're looking for matching funding from any project. This is both academic and SMEs, and ultimately what we're looking for are early stage projects that would otherwise have great difficulty in finding ways to generate their proof of concept data. Again, the IP conditions are, there is no conditions, it's all yours. We have no interest in that, and we're currently assessing a round, and the next round will be in a few months' time in September. I should just say the success rate is around 50%, which I think is probably worth your while thinking about applying. We have had one SME apply in a previous round and was awarded. We've got 2 SMEs in the current round that are under assessment. And the 1.1 million is the value of the total awarded projects. So 50% coming from us, 50% coming from our partner organisations. That's the book, that's the QR code, and again, just to tell you that we can make a difference in not only biomedical, but also in agricultural and environmental areas as well. I think the Kathleen Folbig case is probably the most interesting. You know, the terrible science, the poor science that was brought to that at the early stages. And it wasn't until we were able to bring the sharpest tools, the best minds, and provide the best models that we were able to demonstrate that the mutations that Kathleen's children had could realistically have caused the deaths of her children. And that was the critical change in her case. So again, having the best tools gives us the best data, the best insights, and exposes the truth of the world in which we live. So no matter what you've seen so far in terms of the particular techniques that we provide, whether they're of interest to you or not, I think has already been said by our other NCRIS capabilities, it's the expertise that we're bringing to you. So we have 18 or 20 facilities which are led by 18 and 20 directors. So these are academics, professors who are very much in charge of their craft, their science and their technology, and you have access to them through us. And these are our partner organisations that I said, of which 50% are MRIs and the other half are university departments. So certainly tap that expertise through us or through one of our other fellow NCRIS facilities. And then my final slide is to say tap John. John does send his apologies. He's on his way to San Diego. He's probably there by now at the Bio International Conference. But as Andrew had already said, John is a shared advisor for developing new strategic partnerships with therapeutic innovation and bioplatforms. So I'll leave it there, and I know that we've got some more detail coming in the next presentation about one of our facilities, which is just down the road. Thank you.
Thanks, Michael. So we now have, put my glasses on. Oh, gone wrong. Go back a slide. And we now have Jacek Kolanowski, who's the head of the Research Facilities and Innovation Centre at the Victor Chang Cardiac Research Institute. He's going to talk in a bit more detail about some of the offerings that they have there.
Thank you, Heather, and thank you all of you for being here. Thank you for having me around. So I am actually coming from just next door, so from the precinct of St. Vincent's Hospital, which is in the Darlinghurst. So I'm based at the Institute of Victor Chang Cardiac Research Institute. But what I'm going to talk to you about today is actually the innovation center that we have in there that actually is project agnostic. So that's also very important to know. We are built on the expertise of our researchers, but we are project agnostic in that. And we are, I'll show you also partners of partnering with a lot of NCRIS facilities as well in what we try to do. So I think hopefully it works. Maybe I'll use this one. So yeah, I wanted to just initially acknowledge the traditional owners of country throughout Australia and recognize the continuing connection to lands, waters, and communities, and also pay my respect to Aboriginal and Torres Strait Islander cultures and to elders past and present, and to all Aboriginal and Torres Strait Islanders among us today. So our Innovation Centre is actually something which has been funded heavily by the New South Wales Health with $25 million in 2020, and then we actually, up to now, matched the funding through different sources. So the initial and still one of our main objectives is actually to build the capacity and support capacity in, broadly speaking, cardiovascular research, but both for academia and for industry. But we actually, as I said, we try to combine really in a slightly different way, the originality that can come from academia with a certain robustness that you expect in the commercial space, and also the ease of interaction that we hope we are providing and getting better with every project to work with external users. So we just try to be more and more heavily focused on external users as well. So we are actually composed of 8 different facilities actually. So it's a very broad spectrum, all the way, which allows us to do things all the way from the patient, through the models to the molecule, and then actually back as well. So we can actually understand disease going that way and then redesign the therapies and whatever else and then go back. So we actually have everything from clinical imaging capacities to actually, cryo-EM and all the things across as well. So, and it's all in one space. So I think that's a unique combination that what you can get in there. So the facility that we actually are, which is the partner site for the Phenomics Australia actually, is the stem cell production facility. And again, some of the slides might be quite busy. I will share, the slides will be for sharing after. So that's why I've sometimes put a little bit more text so that you can go back to it and then remind yourself what was there on it. But in principle, what we're doing there, we're developing a more complex models, both from the patients, but also from other sources. So we can develop iPSCs, cell lines, we can modify them with CRISPR, we can differentiate to various tissues, we can grow those 3D tissues as well for you, and we can actually do robust quality control on them. But that's not the end, actually. So we are also phenotyping them quite heavily, and we're able to do that. And so, you know, some of the examples of those models, which Michael has already been mentioning, actually, are in the creation of the research that you can use for screenings, but also development of particular models that you want to test your hypothesis in, but also models in which you can test your therapies in. So that's all kind of in there as well. And I am very, very excited to actually say that we've been funded just recently, again, with the New South Wales government through the NCRIS support program from the chief scientist of New South Wales, with an extra addition to our facility, which is actually a bioreactor systems that allow us now to scale up the production of our models and to make, you know, not few tissues or few tissue kind of constructs, but actually, you know, even hundreds of them for different, and which are quite reproducible for your different applications. So we are very, very happy about this, and this is an effect also of the close collaboration with Phenomics Australia, so we are very grateful for that too. So apart from this model development, as I said, we are actually quite heavy on phenomics, which is a phenotypic characterization, but also providing tools to study those. And we have actually, through the cell function screening, microimaging, and metabolomics facilities, we can have a very robust and also very wide, let's say, characterization happening for your models. So both in a disease understanding, which is maybe less of an interest for the industries, but also in validating the targets, for example, which you want to use in your campaigns, but also actually in just characterizing different variants and in characterizing the responses to therapies. So we have a lot of technologies there, and this is all also supported by the preclinical and clinical imaging capacities in MRI as well, and eco, and also our biomedical data science facility. So importantly, I want to say that we're actually having data scientists there, both bioinformaticians and the software developers that are there for your external use. So this is not just for the research things, but they can you can pretty much just hire those competencies to anything you wanna do in there. And so what we try to do, we actually, as I said, we have quite a comprehensive workflows of infrastructure. The setup in one place is quite unique in Australia. And so through this, you get a one-stop shop opportunity of accessing those things in your research. But we also draw and we constantly adapt new essays from the best articles which are published there. Many people then start to be interested in those essays, but those essays often, as you all know, are pretty specific to a particular model or particular case. So we actually take them up. We develop them into a robust service that you can then come and access. And we do it quicker than normally you would have it in the commercial services thing. So we can really get it in as quickly as that when the things are appearing, and we can very quickly optimize it and prepare it for as a part of the service. And so we really are kind of quite proud and really focused almost sometimes compulsively on certain robustness and reliability. And I think this is something which has always been very close to me. And I really think that we are not doing this enough in the academia, but this is especially important in the industry, I think. And this is really something which we want to be differentiating ourselves in comparison with the other places. But we also actually have an expertise, and this what Audit was talking about, it was actually sounded very familiar, because we try to do it, and this is my role as well, to kind of provide this expertise, or maybe more a consultancy aspect, to discussing with potential users about, even if you know the technology, it's easy, as Audit said, but if you have just a biological question or a problem there to solve, I hope that I can direct you also and propose a certain panel of techniques, both which we can do internally, but we have partners in Europe, also in the US and in other places with the other NCRIS facilities, that we can put you there in contact with those and hopefully design even the whole workflows, or help design the whole workflows for you there. And we also actually, unlike, sometimes you have an experience with academic, I know it because I've been academic myself for a long time, you can have a certain barriers in interacting with academia. So we really want to have the academic-like originality in there, but we really want to work flexibly with you, and we want to kind of really bring it closer to you. So this is just several ways of working with us. So you can actually get trained in self-access. So like with ANTH, we can also do it with other our facilities there. We provide just services, fee-for-service things, and this is very important. We also do not hold any IP on that. So this is with you, stays with you. But we also work in a bit less standard ways in the co-developments, in the validations of technologies also. We actually sometimes pair up with different companies to create joint services as well. We do consulting, as I told you before, but we also co-develop, so we can also kind of like create a service based on your product, for example, that we are sometimes also working with. And this is, the last slide really is about kind of showing few snippets of how we can interact with different partners. And the fact that some examples are at pharma or at biotech doesn't mean that it cannot be done by other sectors or other type of users. But you can see that there is a classic kind of service-based approaches where we allow for the repurposing of leads, for example. So we actually, one of something we are proud of is we can provide you with a panel of the cardiotoxicity assays. We know the cardiotoxicity is one of the biggest issue of benching leads or drug candidates. So we actually can provide you with the very early stage characterization of the cardiotoxicity, extracting a panels that are more predictive than the usual panels that are recommended, and so get the decision-making process easier at the early stages. We also work on the, on everything from sample prep for X-ray crystallography, for example, but also, the standard target validation studies with CRISPR technologies and similar. We actually do a lot of also developing assays, and especially phenotypic imaging-based assays for the early stage mechanism, the convolution, and also more robust picking up the efficacy and the potential also off targets. But we work also with biotech, with IT startups. We actually have been generating data for the IT startups that are in the data, the IA software, or the based software development and similars. But we also work with different types of partners. So if you are selling a reagent or something like this, we co-developed services based on the reagents of different partners. Or we can actually validate your hardware by using our capacity in assay development, assay design to actually show it. And we, for example, were the first one to actually do the functional cardiac MRI of zebrafish. This is not published yet, but this is actually something which we've done a few months ago, first time. We're very excited about this. And we also do protocol optimizations. If you are a CRO, we can also get this, highly published, as I said, assays, turn them into something which is robust enough that you can take and sell. So that's pretty much it. Thank you for your attention, and you can contact me if you want to talk about this through this email, also through the QR code. This is my contact details as well there. And I'm here during the tea break and also later in the networking session as well. And here are also some of our fundings sources, including NCRIS and Phenomics Australia and New South Wales Government. And thank you so much for the attention, and I hope I have a chance to talk to you later about some reading.
TIA, Therapeutic Innovation Australia. I think I'm the last of the NCRIS projects that's speaking today. And our focus is on therapeutic development and research translation of therapeutic therapies. So our aim is to support research infrastructure that accelerates the translation of innovative discoveries along the therapeutic pipeline. As all NCRIS projects are, we're not-for-profit, and we invest government funding in capability and capacity and ultimately to enable access for researchers and SMEs to that research infrastructure. So we're divided into 4 capabilities: biologics and vaccines, small molecule therapeutics, which includes a lot of high throughput screening as well, cell and gene therapies, which is my area of focus, and also recently launching the RNA products capability. We also have vouchers, which Michael Dobby kind of covered a little bit, and ours are quite similar, but I'll give you details about those vouchers and how you can use those vouchers to access the facilities that we support. Again, another TRL pipeline little graph. We sit in that 3 to 6, TRL 3 to 6. So at that research translation, taking your product from the lab, from a research academic environment into something that is suitable for giving to a patient. So a lot of the work that we cover includes kind of target identification, but also the demonstrating efficacy and process development, particularly for the bigger molecule therapies, so the cell and genes and the biologics, but that process development of manufacturing to make sure that your product has a robust manufacturing process. It's also the point, we recognize that it's the point where academic funding trails off, so getting a grant for for academic research trails off at this point, but also the value proposition and your value increases at every TRL that you can increase. And so sitting at this point really helps research and SMEs to get through that valley of death. And that's our aim. So there's a lot of information on this slide, but basically what it's supposed to show is that we cover a cross-section of a lot of therapeutic development services in across different size of molecules. You might see your specific area of need highlighted here, but this is only a selection of some of the services that we offer and that are available. So if you don't see your area of need highlighted here, then reach out and we may be able to know somebody within the TIA facilities that can help. But if we don't, then we might know somebody outside of that, leveraging our network. So this is just a map, similar to Michael's map, of all the different facilities that we support, including the high throughput screening facilities, which we don't support financially, but are available through our voucher scheme. I should mention that there's quite a few facilities in Sydney and in New South Wales that we support, and there are people here today from a couple of those facilities. So we've got Chris and Ellen from the RPF, who are here, and the recombinant products facility. And also we have Dave Rickards from the Biologics Innovation Facility, who is also here. Now, just to go through a little bit of detail about each of the different capabilities that we support. So the small molecule therapeutic capability. This is a really spread across the quite a large part of the development program pipeline, from discovery, hit to lead optimization, and preclinical development. So we have, is that big enough? Yeah. So we have national access to the National Compounds Library for you to do to get products for you to screen your target against a range of compounds. Access to the high throughput screening and other screening, including fragment-based and MS-based screening and assay development for that. Medicinal chemistry, where we can take, where the facilities and the experts in the field can take products and help to generate the IP by tweaking through either computational or synthetic mechanisms to improve and this is probably the only point where IP will be generated and where there might be some IP sharing arrangements with the facility that is providing that assistance. Then there's a lead optimization step to increase the potency and also preclinical testing for so in vivo models that we can have and can develop in combination with the Phenomics Australia as well. The RNA products capability, this is born out of all the excitement that came about through COVID and the mRNA vaccines. Lots of money going into research and into that enabling, into the academic environment, but also lots of companies coming and saying, we're going to build commercial scale and commercial properties here, but what's in the middle? And that's where TIA is sitting itself in the middle, helping to to provide services for developing and manufacturing quality nucleic acids. The biologics and vaccines, so this is where we're now talking a little bit, mostly in that preclinical development point. So we're looking at bioprocess development, scale up of your processes, upstream and downstream development, so that and including your cell lines and optimizing your manufacturing processes, and then also moving into the single-use up to 200-liter bioreactors, and developing your quality control assays as you scale up, so that you've got that quality built in from the beginning. And assuring that you have those you know what the released criteria are going to be, what your quality assurance program looks like. And then the cell and gene therapy capability, which is where I largely sit. And this is, TIA's focus here is mostly on that preclinical development, mostly on manufacturing. So with the cell therapy in particular, the process of manufacturing can often alter the product that you get at the end. And so it's ensuring that you have a reproducible manufacturing process is very important in this instance. A lot of the facilities were reporting to us that researchers thought that they were ready to do a clinical trial but hadn't yet considered all of the things that they needed to consider. So we've introduced a technical feasibility assessment, which is advice to access these facilities that will help you to understand what you need to do in the preclinical space within your own lab to change out maybe some of your raw materials, some of your process development, moving into a closed system, those sorts of things that will be really helpful when you then come to do that tech transfer into the GMP facility for the clinical trial, for example. And all of it is done under a quality. All of the facilities that TIA supports are under some form of quality management system with a TGA or with a FACT or certainly GMP. And so funding. So the Pipeline Accelerator Voucher. This is a way for... So I should start by saying you can actually access the facilities by going directly. You don't need to have a pipeline accelerator to enter the facility. This is just a way to reduce your costs and to get your projects over the starting line. It's a competitive scheme. That my dollars have gone all wobbly, but it's a competitive scheme that provides up to $50,000 of subsidy to access one of the facilities we support. We do need 50% matched funding, and that is cash contribution. It means that essentially the cost to end, you will have to pay half of the value of the work that is done at the facility. It's open to SMEs and academics. We run it twice a year. The next one will be in September, will open in September. Generally, why do you need to apply? So again, you get a quality product at the end. We'll provide you with, the services that are on offer will provide you with proof of concept data just to step up through the next milestone. Hopefully, generally, very few IP conditions. TIA doesn't take any, but sometimes some of the medicinal chemistry may require some IP conditions. We've made 169 awards in the last 5 or so years, and we have a success rate of 56%. Of that, we've kind of tracked a few, and so 48 vouchers that we've been tracking have then gone on to receive $53 million in funding. So it's kind of leveraging the pipeline accelerator in order for bigger and better things, making your product just that little bit more desirable, I guess. It's just a slide of all pretty logos that everybody spends a lot of money making, so should put them up there. If you want to, number 3 of the NCRIS Health Network. But if you want to continue the conversation, you can reach out to me. I'm based here in Sydney. As I say, I'm mostly focused on cell and gene therapies, but come and see me and I can pass you on to whoever, or call me or whatever, and I can pass you on to the right people. Ben is based in Brisbane. He knows everything about bioprocessing that you like, amazing knowledge there. And John, you've heard lots about already, he works for TIA and for Bioplatforms Australia and for Phenomics Australia. So his fingers on lots of pulses, a lot of places. Now, if you can't remember names, faces, you can't remember anything from the previous slides, if you type in TIA into Google, then you'll get some sort of ischemic stroke coming up. But if you type in TIA and Therapeutic, then you will come to TIA's website, and there you will be able to fill out the contact TIA form. We do read them. We are proactive about it. It's not a black box. So please do reach out, and we'll put you in touch with who you need to know and what you need to know.
So now we have a case study on TIA. We've got Nicholas Hunt from EndoAxiom.
Good day, everyone. Thank you all for coming. All for me, I assume, but probably for everybody else. So I am Nicholas Hunt. I'm the Chief Exec of EndoAxiom, and I'm here today to really pay homage to one of the earlier translation grants that we did actually receive. When we started working with Therapeutic Innovations Australia, this was pre-EndoAxiom even existing. This was the first grant that we applied for in a commercial sense, about how we started to bridge the valley of death for our technology. So just as an overview, EndoAxiom was a company that was founded in February last year off the back of some Series A funding with our venture capital meets biotech incubator ProtoAxiom, which is also here in Sydney, New South Wales. And we are focused on the prevention of autoimmune disease and the treatment of diabetes. So what our company does is we develop novel biopharmaceutical pipelines and oral drug delivery technologies, and we take them to clinical validation. So originally we started off as a basic science group in a laboratory. We design nanotechnology transporters for peptide delivery. And the classic one I always talk about is our oral insulin tech. Some of you might have seen was in the news after it got published last month that we take to clinical trials next year. But really we were a technology platform. We were using basic bioscience components, and what we were trying to do is address key challenges around how you deliver therapeutics, how do you make those therapeutics better for patients, and how do you then try and translate those therapeutics to the clinic. So when we first started off with our technology, we were on the back of COVID, which was really exciting because everyone started knowing what a nanoparticle was. And some of you might have received a Pfizer vaccine that has a solid lipid nanoparticle in it. And the key challenge we had when we were trying to design our transporters for nanotech is we didn't use lipid nanoparticles at all. We used other formulations. We used quantum dots, which everyone only really associates as being in televisions, not in that nice one, usually in the high-end range, but they're not used in medicine at all. We also designed our own biosensitive polymers that had never gone through clinical trials before. So effectively we had insulin, which has been used therapeutically before, but a quantum dot and a polymer that had never been tested in a human or any other species before prior to our work. So that really meant trying to go through the valley of death and manufacture that to good clinical practice with nanoparticles that are used in televisions was going to be a really big challenge for us. And that's what our project was really about. We had a product that was high risk. We got advice from both quite large and small pharma companies that we wanted to try and address those challenges. We had advisors that told us exactly the same things. We had grant bodies that also wrote feedback for development grants that this is too risky to be able to try and develop. And within the Australian context, no one had tried to build these before. So we came to CSIRO, probably the largest group at that point that could theoretically even attempt this type of project. They said it was not impossible, it would be very much a challenge, but we needed to start from the beginning. We could make this in a lab, but could they even reproduce it itself? And what we came to TTRA to do was to try and get some initial funding to just do some pre-examination about could anyone else even build this and could it be therapeutically valid. So this branches into why we partnered with CSIRO. We had no internal capability to manufacture these materials ourselves. So we were based out at Sydney University in the Sydney Local Health District. They are now building a brand new shiny SBA building that will have GMP capabilities, but at that point in time that didn't exist. But there was also a very big drive about advanced manufacturing and building the next generation of technology. And this was something that the government was strongly pushing for. And the idea if we could build nanotechnology platforms that were the next advancement in nanotech options, then you might be able to attract a level of funding. And of course, we're also attracted to the shiny check that also came with it, as long as we can contribute half the money. So what was the actual project? I'm glad to say this is the end result of what it did actually look like. But effectively what we were trying to do is conjugate quantum dots to a protein, encapsulate it in a polymer that we'd purposely designed to be reactive to a biological environment. We built this particular polymer because it will react with enzymes that are in your blood system. So if we have an oral insulin that we're going to give to a patient, the enzyme will react to, the polymer will react to enzymes in your blood that are dependent on how much glucose is in your blood. So it's a smart sensing oral insulin tech, which is really cool from a scientific point of view and trying to publish it, but not so much when you're trying to get someone else to try and manufacture it from a GMP standpoint. So this brought together a whole bunch of expertise across university and medical school, but also our business school about how we can start developing a business plan for this. But then to also go for bringing together clinicians about what they actually saw the challenges for this tech were, but also bringing together Therapeutic Australia and also CSIRO. So what the outcomes for this project were, and needless to say, now that we have a company, I'd like to say we were successful, is we managed to build the first R&D blueprint for this tech from an R&D point of view. That then led to a second contract with CSIRO to do the non-GMP manufacturing, and now we're up to the next rendition of that, where we're doing GMP manufacturing for the first time in Australia for this type of technology, and also around the world. Speaking to other nanotechnology companies within Australia, they do their manufacturing overseas. So the ability to even do this within Australia was incredibly exciting. What we also got is external validation that we could build these materials by another lab group, which also added to the ability to actually publish it in quite an esteemed journal in Nature Nanotechnology, which is really exciting. But what really the main outcome was, is we've been able to de-risk this tech. When we're applying for grant systems, they told us the challenges are going to be, can this even be done? So on the back of this grant, we were then successful with an MRFF TTRA grant. And the first milestone that we had to show was successful was this project. And that then unlocked the funding for that particular grant. Off the back of that, we then attracted Series A funding in a flow-on effect after receiving that MRFF grant. So really, it's a snowballing effect. De-risk your largest problem, and then funding does continue to follow. And that's it. Thank you.
So now we're going to hear from the National Measurement Institute, starting with Stephanie.
Well, hello everyone, and good afternoon. Thank you very much for being here today. It is truly a pleasure to have a moment to speak of a very interesting topic, which is fundamental to science, industry, and everyday life, and that's metrology. Whether you are directly aware of it or not, metrology makes many of our daily activities possible, from brewing our morning cup of coffee to determining the radiation dosage to cancer. Metrology principles are all around us. I'd like to start with a brief history of metrology. Metrology, the science of measurement, has ancient roots. From the early use of body parts, as we can see that happened in Egypt, units of measure, to the precise standards of today. The evolution of metrology reflects our continuous quest for accuracy and reliability. Throughout history, civilization has developed systems of measurements to support trade, construction, and science. The advent of the French Revolution made a significant milestone, paving the way for international standards that facilitated global collaboration and innovation. An essential aspect of our work involves legal metrology. Legal metrology ensures the accuracy and reliability of measurement using trade, health, safety, and the environment. It encompasses regulations and standards that protect consumers and maintain fair practices in the marketplace. It's very interesting when we merge metrology and legal metrology, which these two terminologies create confidence in what we do. So the National Measurement Institute. Our institute, the NMI as we call it, is a division of the Department of Industry, Science and Resources. And we are responsible of measurement as described in the Measurement Act of 1960. Our work spans a wide range of activities involving the development of new measurement techniques, the calibration of instruments, and the maintenance of national standards. We benefit industry, the community, and the Commonwealth, delivering high accuracy measurements and help ensure emerging markets needs are being met. So this is a very interesting slide because incredibly, we also do trade measurement where we ensure that, for example, the meat that we're purchasing in supermarkets or the kilogram of the tomatoes that we're actually purchasing are actually the kilogram that they're saying that it is in the balance, in the scales. We also, for example, ensure that the petrol, like the amount of gas that goes into the airplanes, it's actually the amount of gasoline that the airplanes need. Otherwise, we would be measuring wrong. So the National Measurement Institute is divided mainly into 4 branches. And this is the Chemical and Biological branch, Physical Metrology branch, Analytical Services branch, and the Legal Metrology branch. And within these 4 branches, we have many scope. I could take days explaining all of the capabilities that we do have, but Our capacities or capabilities range from providing certified reference materials to proficiency testing, to chemical and biological certified reference materials as well, to the capabilities of measuring or having a nanometrology laboratory, to training, and also everything that concerns trade measurement services. Our state-of-the-art laboratories in Sydney, and which we have two in Sydney actually, one in Linfield and the other one at North Ryde, and our other state-of-the-art laboratory in Melbourne, and our set of expert staff, we are designated to meet the highest quality of accuracy and reliability. The NMI, it's a great partner for companies in the research and development space for several reasons. And we like to show this slide because we actually sit up in the middle of the whole journey of bringing an idea to the actual market. So in the National Measurement Institute, we're gonna be able to help you out with prototyping, with testing, with validation. We also have the laboratories and the facilities to be able to do this. And we will also be able to help you out with your quality assurance and process optimization to be able to create standardization at the same time. And also, we will ensure that your product complies under regulation, so you can actually be accepted globally. I'm going to delve a little bit into the technical expertise that we can offer across the therapeutics field. This is super, at least that it's never ending. So I'm gonna be quite brief on this one, but in terms of our technical and scientific expertise, we can do calibration to ensure quality control, and this extends into medical devices as well. We can also do pharmaceutical testing within, for example, testing for trace elements in pharmaceuticals or allergens, for example. So part of the National Measurement Institute and the Analytical Services branch has the capacity for a fee-for-service to test many different contaminants, for example, or in terms of the calibrations of critical equipment, some examples are calibrations of lasers and energy meters, ultrasound equipment, and this is kind of the area of our expertise within the chemical and biological branch, which might be quite relevant to this, to all of you guys. So our biological section can specialize or specializes in genetics. So we use digital PCR and mass spectrometry to provide traceable genetic measurements. And we also provide testing services in different areas, such as cancer diagnostics, animal feed species identification, for example. And we also provision calibration standards for genetic measurements. We also have the capabilities for RNA. So we can do custom RNA reference materials with concentrations that are traceable to the international SI unit, so they can be accepted globally again. We measure our RNA concentration directly using isotope dilution mass spectrometry. And we can also, or we are also working actually on the identification or characterization of lipid nanoparticles used in the delivery of mRNA vaccines. So this last part at the end of the slide, what it's saying is that we also have the capability to accurately measure the amount of the process of reverse transcriptase to be able to give a very accurate measurement. Part of the process of COVID, of our pandemic from COVID, we are preparing in case another pandemic arrives. And what we're doing across this area is we are producing high quality controls for digital PCR for COVID. We're also working on nucleic acid measurements for rapid antigen tests. And we also have acquired a newly oligonucleotide synthesizer for the producer possibly of primers and probes. And we are actually developing high quality control materials for digital PCR assays for organisms of concern such as influenza and H5N1. In these photographs, you'll be able to see our nanoparticle characterization laboratory. This is located at Linfield, and this is a perfect space with our team that has more than 30 years of expertise in particle characterization. There's more than 17 instruments and a specialized AFN facility for precision dimensional measurement. And the interesting thing about this facility is that we also hire it in case you guys want to come and do your research at our facility. So some examples that we have done with the therapeutic space and nanometrology has been working with universities within the lipid nanoparticle area for vaccines. We have also characterized particles, for example, in sunscreen. And we have also worked with companies developing APIs based on microparticles to characterize the particles and try to narrow down the size distribution and increase the effectiveness of the product. So some of the recent projects that we have been collaborating within industry, it's the calibration of sensors at -80 degrees for vaccine storage. We have also been working on the production and validation of genetic standards and quantification methods for clinical manufacturing of gene therapy drugs. And our beautiful people, which is Sharon and Michael, who are joining us today from the biomanufacturing facility at Westmead, are joining us today to explain a little bit more in detail how we have worked together. Another area that we have been working with industry, it's the monitoring and detection of pharmaceuticals and contaminants of emerging concern in waterways. Last but not the least, we also provide industry training. One of our training courses that it's the most famous one that we have, it's the measurement uncertainty. And we also value our student programs quite dearly, providing summer vacations, internships, honors, masters, and PhD projects. So I hope in this nutshell, we have given you a brief overview of the National Measurement Institute, and please do not doubt to contact us if you have any measurement concern or you need us. Thank you so much.
Okay, hi everybody. Thank you for the invitation to speak today. I'm a scientist in the process development analytics team with the New South Wales Viral Vector Manufacturing Facility, and I'm here with Michael Shun, who is the project manager. So this facility is newly formed, formerly the Sydney Cell and Gene Therapies Group. And so it's an exciting new initiative that's been supported by the New South Wales Government and Sydney Children's Hospital Network. And the purpose is for the manufacturing of clinical grade viral vectors for cell and gene therapies on home soil. And we are at Westmead, located within the Kids Research Institute. And we're here today to tell you a little bit about what we're doing and also how the National Measurement Institute team are playing an invaluable supporting role in this journey with regards to developing product-specific and process-specific analytical toolkits, which are going to benefit greatly in terms of cost, time, and result in a fit-to-purpose product. As you know, acquiring commercial kits from overseas are very expensive, takes a lot of time to get these kits in, and they're fairly generic in their purpose. Okay, so cell and gene therapies treat disease by introducing genetic material into a patient's cells to repair or replace faulty genes and introduce novel functions for curative purposes. And gene delivery vehicles or vectors can be either viral or non-viral. And adeno-associated virus and lentiviral vectors are the leading, most widely used viral vector technologies in clinical applications of gene therapy, and these are the two viral vectors that we are going to be focusing on in our manufacturing. So AAV vectors are mostly used for in vivo gene transfer, where the product is injected directly into the patient, and currently the most well-known treatment or gene therapies for AAV are to treat spinal muscular atrophy and retinal disease. And lentiviral vectors are mostly used for ex vivo gene transfer, where the cells are taken from the patient and the viral vector is delivered to the cells outside the patient and then delivered back into the patient. And so together these AAV and LVs represent around 70% in each category of current clinical trials. And the mode of mechanism of the particular vector and also the gene delivery method influence very much the analytical tests that are required on each of these products. So it's estimated that more than 65% of cell and gene therapy manufacturing is currently outsourced to contract development and manufacturing organisations, and we actually have experienced having to have a clinical product made and had to go offshore to the US, and there's waiting periods of up to 2 years to get into the queue. As manufacturing capacity is at least 1 to 2 orders of magnitude lower than what's currently needed to support current and future requirements. And even the FDA has recognised even further increase in the products that are going to be produced. They expect that 10 to 20 products are going to be needing approval a year by 2025. So there's been a big push for development and manufacturing of gene therapies on home soil on the Westmead campus since 2003 under the leadership of Professor Ian Alexander. He set up a small clean room facility to support small-scale research and development, and this has eventually led to the building of a dedicated facility to support manufacture of clinical-grade viral vectors on a larger scale. So big efforts from Ian, along with Leszek Lasowski, who is Vector Genome and Engineering Head at CMRI. So they have been instrumental in acquiring funding for this initiative. It started with a $2 million grant from the Office of Health and Medical Research in 2018, which led to facility upgrades and purchase of production equipment for up to 25-litre capacity suspension cultures, and then more recently acquired a $25 million grant from New South Wales Health to take us to stage 2, which is in construction and will consist of a large-scale production facility at the lower ground of the Westmead Health and Innovation Centre, and also construction of process development and quality control labs. So the viral vector manufacturing processes are separated into 2 phases. So upstream manufacturing involves transfecting producer cells with plasmids and harvesting the viral vector produced, and downstream processing prepares the vector for administration to patients. And all along the process, there needs to be monitoring of quality attributes of the sample in order to optimize the processes, and also on the final sample for release criteria. So the critical quality attributes for gene therapy products basically fall into these 5 categories. And so we have had immense help from the team at NMI on one of these categories that was causing us particular, we were struggling with, so purity. So this is very, very important aspect from the process, from the manufacturing perspective, and also for safety on the final product. It looks at any residual plasmids and host genome DNA in the sample, and also size, fragments of DNA size. So the formal relationship that we have between the team at NMI and the VVMF really began only 6 months ago, although the relationship with Westmead extends far beyond this. And in this very short period of time, the team have put in an incredible amount of effort developing validated PCR assays and nucleic acid standards to help with quality control in the GMP production of viral gene therapy vectors. And specifically, they've been focusing on analytical tests to detect residual genomic DNA from the host cells used in vector production, and to detect, to quantitate and measure DNA fragment size of residual vector plasmids and host cells, along the lines of what Stephanie described in her presentation. And this is using cutting-edge technologies, quantitative PCR and digital PCR technology, and we are currently using these tests on our in-process samples for optimizing our lentiviral production, which is the viral vector we're starting our manufacturing journey with, and this will also be applicable to our AAV manufacturing pipeline. The other thing that's very important, especially on the final product, is strength of the product. So looking at the infectious and genome titer and biological activity. And so future work with NMI, and also some of this will be a continuation of previous projects commenced, we'll be looking at particle quantification and potency of the product, again using PCR technology. And so specifically quantifying lentiviral, proviral DNA integrated into the host genome, so that's a measure of potency. And then when we come to the AAV work, we'll be looking at particle titers, using a whole lot of other tools that NMI have developed. And basically that comes to my last slide. So I would just like to acknowledge the team at NMI, so Daniel, Anna and Jacob, who have been working very, very hard on developing these toolkits for us, and also the team at the viral vector manufacturing facility that have been directly involved. So Ian Alexander, Leszek Lesowski for getting this off the ground, Michael, myself, Ken and Kunal.
So now we're on to our very last facility. So we're going to hear from Gina at ANSTO, and then their case study is straight after that.
Okay, last but not least, I hope. All right, first things first, because it's the last one, let's just give a bit of a stretch. If you ever heard of ANSTO, raise your hand. Raise your hand higher, because you want a bit of a stretch. Now, if you never heard of ANSTO, raise your hand. That way I cover both sides. So most people actually heard of ANSTO, that's great. All right, let's try to work this out. Okay, how does this work? What? No, okay. Right, okay, I was just getting used to this. Okay, so ANSTO is Australian Nuclear Science and Technology Organisations. That's what ANSTO is for. Our mission is to deliver knowledge, value and trust through the applications of nuclear science, technology and engineering. So ANSTO is here for you. We are federally funded government, so we'll see. So our objective is to deliver on Australia's priorities for the benefit of people, industry and the environment. So for the people, again, that's you. Improve the health of Australians by supporting access to current and future nuclear technologies, and to be Australia's source for nuclear expertise, advice and services to government, academia, industry and community. So this is ANSTO. How do we actually do that? So we are a leader in nuclear science and technology. Yes, I'm going to go for a bit of a walk because I do need a bit of a stretch. We have operated safely for over 70 years. In fact, last year we just had our 75 years anniversary, which is pretty cool. We have over 1,350 skilled employees over 2 sites, which is in Lucas Heights. Have you guys been there? No, raise your hands if you've been there. That's perfect. That's a lot of people. That's awesome. I'm actually quite surprised and happy about it. And the other side is in Clayton campus, which is in Melbourne, where we house our synchrotron. Okay. I actually have to walk around here and there. So we own quite a lot of national research infrastructure, as you can see. So to me, my colleague there was actually kind of complaining why you only have 15 minutes because you have 1, 2, 3 facilities funded by NCRIS, and we also have our Australian synchrotron, which is funded by National Innovations and Science Agenda. So I'm trying to do this like a speed dating because it's a lot to cover. So our facilities and capabilities. Let's start with Opal. Opal is a lovely, lovely, lovely reactor. Okay, I'm just going through this because I actually have my notes on this, which is a bit of a pain. So it's an Opal pool Australian light water. It opened in 2007 and it uses low enriched uranium. So it's not for generating electricity. It is actually a multi-purpose reactor. The main part of it is for neutron irradiations. So we do silicon irradiations to make the silicon ingots becoming a good material for a semiconductor. And using our neutron irradiations, we also produce this radioisotope. So the generator that you see in hospitals for technetium-99m, yes, we make those generator on site. And on top of it, we generate neutrons for Australian scientists. How? By this thing, neutron scattering. So we have Australian Centre for Neutron Scattering, or ACNS. You might have heard about it before. Yes, it's an NCRIS-funded facility. And it uses neutron beams that we got from the reactor for all areas of physical and life sciences. So when you look at it, what is the applications of it? So the applications of this particular infrastructure is that you can actually use the beam to look at structural analysis for biological molecules and structures. So for example, if you're interested about, it's actually starting from a macro structure. So if you're looking at a stress test on the train tracks, you can actually use our facility. But we can also look at very micro structures such as protein or your molecules, even the structures of the molecules and how molecules actually interact. We can use our facilities to look at that. And we also use powder diffractions to also detect trace of elements in your samples if you're looking at, purity, for example. So on top of it, now, because the neutron scattering uses neutron, they're very, very handy for deuterations. Now, has anyone heard of deuterations? Ah, perfect. 1, 2, 3, 4. Great. Awesome, I don't need to talk to you about this then, no, forget it. So deuterations. So deuterations, what happened is we added neutron into the molecule. Now, the beauty about these deuterations is that the characteristics of that molecule is not changed. Okay, the structure is not changed, it's just heavier. So we custom made deuterated materials, and yes, it's another NCRIS funded facility. So what we do is we provide deuterated molecules to researchers and industry. We do chemical and biological isotopic labeling, and we help you with investigate your structures really. So we have unique expertise and specialized lab space and equipment and awesome people like Tamim. Okay, so what's the use of this? As I said before, it's really, really handy when you have neutron scattering facilities right next to us. So these deuterations actually provide contrast in neutron scattering experiments. So you can actually see with neutron scattering, because we have extra neutron, you can see it in your experiment. So if you're interested about phospholipid, like different kind of lipids, and how it interacts, or how it changes in your molecules, you can actually label that with deuterations. And then you can actually see how it interacts in the membrane, or how it actually gets metabolized. You can also see the way deuterated molecules work. It also spins differently. So you can use that as a contrast with the NMR. So who uses NMR here? I love you people. And we can use deuterations with the mass spec for very similar things. So if you wanted to know the metabolites, you wanted to know how your molecule works, you can actually use mass spec. In fact, our lovely next guest, which is Amy, is going to talk about how we use deuterations in the internal standard for mass spec. And on top of it, deuterations can improve the metabolic stability of drugs and radio tracers. So we're looking at, we wanted to use small amount of molecules for a big amount of effect. So we look at cosmetic, we're looking at drugs. So if you have a partner, if you're keen to partner with us, here we are. All right, next one, our facilities and capabilities in radio labeling and radio imaging. Yeah, we play with a lot of radioisotopes. And because of that, we have quite a big array of license in the use of radioisotopes. We kind of wanted to focus a little bit more on the radioisotopes that we produce from our reactor. So we utilize hot cells and safe handling methods for our materials. And on top of it, to know where they're going, we have our preclinical imaging capabilities, which includes PET-CT and SPECT-CT, and as well as autoradiography. So what is the applications of that for you? So what it means is that if you do have a materials that you wanted to know where it goes when you administer it to a mouse or a rat or even a human, you can actually put a radio tracer in that, inject it, and using PET-CT, you can actually see where it goes. Does that make sense? I told you I love you guys. All right, next one. I'm just 8.5 minutes. Yes, I'm watching the time, Elizabeth. Okay, the next one is about irradiations. Now, do any of you guys know that we do irradiations down in ANSTO? No, okay, this is new. So we house a gamma irradiation chamber utilizing cobalt-60. So it has 10 milligray to 1 megagray, and we also use that for precision irradiation. So the applications of it, so when you irradiate something, you can sterilize them, okay? So we can actually sterilize. If you have a product that you wanted to insert into human patients or really use them in a clinical setting or even preclinical, you wanted to make sure that they're safe, that they're sterile, we can actually sterilize it for you. So if you're in the early stage of development, we can help you with the sterility part of it. So we actually know what dose do you actually need. We can also look at the impact of irradiation. So, you heard of things where you're using a therapy using radiations. We help universities looking into the impact of those radiations in human and animals, actually. And we do therapeutic material development. So we're currently working with a company developing vaccines using a irradiated bacteria, which is actually going pretty good because it shows that the memory or the learning from that particular vaccines transferred into a different strain, which is great. And if you're interested, if you wanted to go to the space or if you wanted to send your stuff to the space, we do the space readiness test for you because we can mimic the irradiations that you get on the space. So there you go. All right. So next one is we have an accelerator, which is Center for Accelerator Science. Maybe I should go like, anytime you see NCRIS, you go like this. And I'm so sorry, I do not have the graph that you guys have. You know that. You go, you're going to see this so many times. So CAS, Center for Accelerator Science, it operates for ion accelerators with an energy range from 0 to 10 megavolt. So the applications of it is mainly in environment. So environment and as well as carbon dating. So not so much on the health sector per se, but what you might be interested is this, characterizing trace elements. So we are currently working with industry looking at PFAS. So, you know, PFAS, we don't like PFAS because now we understand the danger of PFAS, the hazard of PFAS. So what we do is we use the accelerator to look at the presence of fluorine in food packaging to know, you know, the presence of PFAS. And last but not least is our lovely synchrotron down in Melbourne. No, this is not NCRIS, this is something else. So we just recently putting a lovely solars there. It's a lot of solars, but it's pretty cool. It's pretty handy. Now, what is a synchrotron? I actually got my colleague who is a physics to explain to me. No, I kind of know what it is, but the way he explained to me is like, it's hold on, I'm just going to do this with my thing. So synchrotron produce intense beam of light ranging from infrared to X-ray. Okay, so you can imagine this is your synchrotron there, and then you have that beam of ray of light going through around in a circle like a ring. And then out of some parts of that ring, we got a bit of an instrument to catch them. So the applications of it is elemental, structural, and chemical analysis for crystalline, molecular, and protein, and as well as X-ray imaging. So elemental imaging, so if you're interested to know what is inside your materials, which is quite useful for medtech, especially if you wanted to know, like, you know, even the surface element, we can do that. We can also do a CT, which is really, really nice because it doesn't really take that much longer. And also you get a really good resolutions compared to what you would see somewhere else, because again, there's only one synchrotron in Australia, and that's us. All right, so if you are, actually I'm going to ask Odette, I think with the ANFF, maybe if, for example, a lot of people doing a 3D printing nowadays, so we can help you analyze, is the 3D printing that you get is similar to your design at the beginning? Okay, I only have 1.5 minutes. Okay, well, I work with ANSTO. If by now I haven't told you how awesome is our research infrastructure, I hope I did. At least I can tell you we have the know-how. So our research scientists, they're amazing. They're very, very knowledgeable. They're expert with their instruments. And on top of it, they're really, really friendly and they're really enthusiastic, and I really love working with them. And on top of it, if you are working with us, we will provide you with training, because it's not every day you're going to play with these lovely instruments, and they are hard to use. So how to access us? Now, if you ask us, I would love to give you fun, but we're kind of strapped for cash, so no. But how to access us? Through merit. So our user proposal opens mid-March in mid-September. So if you have IP that you wanted to retain, this is probably not for you because it's non-proprietary. And we do expect that if you have a publication, it'll be a co-authorship on the paper. It's also quite competitive, by the way. Or you can do halfway, kind of pay some part, and we pay some other. Or you can do commercial way, where we will help you going through the whole project, but you do have to pay for that 100%. Or if you are using NDF, the iteration facility, yes, I'm going to promote that now. As earlier said by Heather, yes, we are part, and Michael, sorry, yes, we are part of the voucher scheme that they have, and the Phenomics and TIA. So if you're keen to use the two iterations, then absolutely I would encourage you to do that. Last and again, not least, I hope you enjoy the talk and I hope it makes sense. So this is our capabilities. Just give me an email, yell out, and it's actually my KPI. If you email me and say I did a good job, that's awesome too. So yes, just QR it and it actually goes to my email straight away. So yeah, thank you so much. And I'm actually looking forward to you, Amy, but yeah, thank you.
Thank you. Love the energy.
All right, tough act to follow, actually. I'm Amy. Thank you for still being here, close to 5. I'm from BCal Diagnostic, and I'm the Product Development Scientific Lead, fancy name. But at my core, I'm really a lipidomics mass spec scientist. So does anyone know mass spec here? I'm assuming some people. Yep, perfect. So at BCal, we really aim to transform breast cancer outcomes. So we've been developing a blood test for early detection of breast cancer. And today I'm going to talk a little bit on our product development pipeline and also our collaboration with ANSTO, National Deuteration Facility. Now, needless to say, breast cancer is the most common cancer among women. 1 in 7 women will develop breast cancer in their lifetime. Now, the current screening method really centers around 2D mammography imaging technique. Now, this is an old imaging technique. It has its flaws. It's not perfect. For younger women, especially if you have higher breast density, the detection rate actually is pretty poor. Now, not to mention, it's extremely painful and uncomfortable methods of imaging. I wouldn't wish that on all men, hopefully. You won't have to experience that. Because of that, it's very prone to self-exclusion. And in other countries, there are also cultural reasons that women won't go for screening. Now, we know that early detection of breast cancer will improve the survivability of these patients, so the earlier the better. Despite having a national breast screen program in Australia, which we are all really lucky to have, most countries do not have that. According to their report, only about close to 50% of women will go for screening. Now, for the other half, whether it's access issues or if it's because of age, not sure. Now, at BCal, we're focusing on using lipids as our biomarker. Now, lipids, if anyone knows lipids here, I know Tamim does, great. Now, lipids are fundamental components of our cells, so think of it in your plasma membrane, as we all learn in school. It's also been shown to be involved in multiple cancer signaling processes. Now, lipidomics using for detecting the disease biomarker has been around for ages, and you will read a lot of literature about it as well, but to take it into translation into a clinic, that's not easy. Now, at BCal, we are measuring the levels of lipids in the blood plasma of these patients, and we've developed a machine learning algorithm and looking at all these different lipids levels for early detection. Now, when I said earlier it's not easy, now lipidomics is not for the faint of heart. I can tell you that because I do it daily. Now, if you look on your right, blood plasma lipids really cover a broad range. So it goes from millimolar all the way down to picomolar. So what that means analytically, it's very difficult to measure in one go, depending on what lipid biomarkers you have. Now, on the structure itself, if you look at that pyramid, the elucidation of lipid biomarker structure actually impact its identification and also then the synthesis of the standards. Now, we use a wide range of mass spectrometry techniques to actually measure those and identify our markers. But now when it comes to going into clinical, you will go into validation and you need to actually go into a targeted approach. That makes its quantification very crucial, and you need to go into its accuracy in quantification as well. For that, we require high quality and reliable reference standards and also stable isotope label internal standards. Now, globally, there is a shortage for such standards, especially in lipids. It is quite a niche. We do work with offshore commercial vendors that have such capability, but then they are offshore, and there are really only 2. And then when you talk about standards, and I might touch on that a little bit, there are different grades of standards. So going from most rudimentary research all the way to a certified reference material, and all that involves multiple different processes of verification. Now, our work with ANSTO, the National Deuteration Facility, has been really great. That really helped us progress into our preclinical development work. And I'm just going to talk a little bit about the process and our relationship working with NDF here. Now, I'm a scientist. I don't do much business talk. But when I work with commercial entity, offshore especially, very often I get linked to a business development associate. This is business talk first. As a scientist, I would like to talk details, but I can't talk details. It's possible, but it takes so many rounds of communication back and forth, and also for them to contact their internal team. That process is very often too long. And for very challenging lipids, which you want to customize, it's best if you can talk direct to the scientist. So not only approaching it from a clinical or biological sense, looking from a blood plasma perspective, but also from a chemist perspective, what sort of processes is required for such synthesis. So I really appreciate the direct communication and to start this synthesis project with Tamim and his team. And yes, like Gita said, they are really nice guys, and not, yes, scientists and chemists, but not weird. Now, just a little bit showcase on one of our earlier work here. So in partnership with NDF, synthesizing some of our custom lipid biomarkers, and also our US lab Pression, we actually developed an assay to quantitatively measure these lipid biomarkers. Another note for our work is also lipids are tricky. So using biobank samples sometimes can compromise your data. You're not actually sure whether the signature you're looking for are artifact for storage from biobank, a different collection procedure for the primary blood collection tube. So the majority of our samples are all prospectively collected according to our criteria. And we have expanded collection into the US, India, Europe, and currently in Australia, we're also recruiting any participant who is willing to donate. And by measuring all these lipids, we are slowly narrowing down into our final lipid signature, and we are in the process of actually developing our final assay, which we will take into validation. Now, I'm quick, lucky last, I won't stop you from going for networking drinks. Now, transforming breast cancer outcomes, a call for collaboration and acceleration. Now, our path from discovery research to clinical translation for using lipid biomarkers has really not been straightforward. We encounter a lot of technical challenges and also challenges with samples and access. Now, our experience also highlights the importance of collaboration and having onshore support like ANSTO, NDF. It really helped us mitigate the risk of dependency on using only offshore supplier. It's not to say you cannot, it's just that from our experience, there have been a lot of logistic issues as well. As you know, Australia is very, very far away from the rest of the world. So this is a call for continuous support for fundings to increase capacity and also to guarantee time for medtech like us and researchers to be able to access Australian leading facilities like ANSTO's and others today. And at the moment, we're also seeking opportunity to scale up our synthesis effort. So we'll be looking for grant funding opportunities. And we're also trying to take our lipid standard to the next level in accrediting them according to various ISO standards. Now, lastly, I'll just, as I have time, I'll just talk a little bit on my experience. So I'm a mass spec scientist. I come from an academic background, went into clinical trials, then gone into pathology. And now I'm in commercial. So I feel like I've gone full circle. And really, in each of the industry I've been in, there are a lot of silos. So people don't talk to each other enough. And very often, a lot of the problems are quite simple to solve. But if you're focused on your project for such a long time, you ended up just not looking elsewhere. And I find the easiest approach is just to ask. So my story of the day is to encourage everyone to just talk to each other. It can be a very stupid question and you might embarrass yourself, but that's okay, because tomorrow you're probably going to forget about it. And that's just an acknowledgement for all our partners, all the entities that we work with, and we also have a strong affiliations and partnership with University of Sydney, Preston in the US, and we're starting our clinical studies all over Australia, and some of these are brand new logos. Thank you so much, and thank you, Ansto and Tamim as well. If you guys are looking for any collaboration, projects, or questions, please come and talk to us, and we'll be around. Thank you.
Thank you, everyone, for sticking with us for so long. It's great to see that you're all still here. I have to say, Geeta, I'm a little intrigued that Ansto's been around for 75 years and operating safely for 70. Maybe we won't manage. Maybe we shouldn't mention that. We did go out to Ansto yesterday and did a site tour. Thank you, Geeta. And one of the amazing things, as Elizabeth pointed out, was you just don't know what you don't know. And it wasn't until we went out and saw everything that they do out there that we really got to understand that a bit better. And hopefully what you've got from our presentations today is a better understanding of what you didn't know about all of the facilities that exist to help you out. If your head is spinning and you don't know where to go, you don't know what to use, or there is some kind of widget that you need that you didn't see in the presentations, MTPConnect exists to help you find those groups. We're friends with everyone who has presented today. We know them well. We know CSIRO. We know the companies in the space. We're an independent organisation, so we don't have to point you to members. We want to help you find whatever is most useful. Success for us is helping you find what you need to do what you need to do. So with that, I will say thank you again to all of our brilliant organisations who presented today. A huge thank you to all of the companies who took the time to come and talk to you about what they've gotten out of working with these facilities, and invite you to go and have a drink and some food. Thanks, everyone.